Good afternoon and welcome to this PHONZ live presentation and seminar titled Errors and Misconduct in Biomedical Research. My name's Peter Hemona. I'm a committee member of PHONZ. I'm going to start us off with a karakia, acknowledge those past and present, and then I'll introduce our speaker today, Elisabeth Bik. Kia hora te marino kia papapaunamu te mona hei hurei mā tātou I tēnei rā. Aro mai tātou I tātou, home e hui e tāki e. Te hau kainga, tēnā koutou, rātou kua wihi ki te pō, moi mai rā rātou. Rātou kia rātou, tātou kia tātou, tēnā koutou, tēnā koutou, tēnā koutou katoa. PHONZ is very pleased to introduce our speaker today. It's Dr. Elisabeth Bik. She is a Dutch-American biologist who has worked for 15 years at Stanford University in the last 2 years in industry. Since 2019, she has been a science integrity consultant at Harper's Bik LLC. She scans medical literature for images and other data of concern. She has found over 9,000 problematic scientific papers and has resulted in over 1,000 retractions and another 1,000 corrections. For work on exposing threats to research integrity, she received a 2001 John Maddox Prize and a 2004 Einstein Foundation Award. Without further ado, I'd like to introduce Elisabeth.
Thank you so much. Yeah, and thank you for for hosting me here today. It's my absolute pleasure to talk to you about my work. And so I was a microbiologist at Stanford in around 2013, and I'll talk about how I got into this somewhat slightly different career, quitting my job as a microbiologist. But before I start my talk, I want to give all my my conflicts of interest, I guess, because I did quit my job. And so people are always interested in how I make my money. And that is by giving talks or by consulting work for publishers or for institutions or funders, scientific funders who have received allegations of misconduct. And I might work for them quickly, like a short period, to investigate those allegations. I also get donations through Patreon, so people support my work, because there's not a lot of grants for science integrity work. There's a lot of grants to do science, but not to really do the checks and balances that I feel are necessary for science. And I also get free access from Proofik and ImageTwin to access their software to help me find duplicated images. I also am a Stanford Metrics affiliate. So Stanford Metrics is the meta science branch of Stanford University in San Francisco, near San Francisco, California. And by having that affiliateship, which is not, it's not an employment, but I do have access to the Stanford Library. So I can have access online to the scientific literature, which obviously is very much needed for my work. So yeah, those are all my financial disclosures. Okay, which button to press? It's not the arrow button. Okay, maybe I should do this. Oh, thank you. That's different from a Mac. I'm a Mac person, so I'm always lost with these keywords. Thank you. So how did I start my career? I worked at Stanford, and I first heard about plagiarism. Well, I knew what plagiarism was, but some podcast or so I was listening to was about plagiarism, and I wanted to know if somebody had plagiarized my work, just for shits and giggles, as we say in the US. Like, I wanted to just take a sentence I had written, put that into Google Scholar between quotes, and then see if something else came up other than my own paper. And lo and behold, a paper came up, an online book chapter that had the text marked in red, which was my text, but somebody else had put their name on top of it, and also had stolen other sentences and paragraphs from other scientists. So on the left, you see this colored text. The text in red is my text. The text in green and purple is text by other folks. It was this patchwork of plagiarized text. And what I felt that moment was rage, was anger. I was so angry that somebody had stolen one of my sentences. And that anger is still fueling me today, because this was not science, this is dishonesty. And so I also started working then on plagiarism for a while as a hobby. I was still fully employed. I worked on the microbiome of humans and dolphins at Stanford. But in the evening times and in the weekends, I would search for plagiarized text. And then one day I came across a PhD thesis with a plagiarized introduction. So the person who had put their name on the thesis had also stolen text. But they also had problems with their images. So I noticed as I was flipping through the PDF that there was this one particular image of a Western blot. That is the image you can see at the middle of the screen at the bottom. I've marked it with a cyan or turquoise box. This is a Western blot. Now, I assume not everybody is familiar with Western blots. I know one person who's very familiar in this room, but this is basically what you need to understand. These are protein blots, but they're basically horizontal dark stripes on a light background. I think everybody can see that. And hopefully you can also see that there's one dot above the fourth lane, as we call it, the fourth band. Now that dot sort of, yeah, I recognize that because as I was flipping through the chapters in this PhD thesis, I saw the same photo, what you can see on the bottom right. It's the same photo, except the photo has been mirrored and it's a little bit cropped differently. But it's the same 4 lanes, the same 4 horizontal bands and that little dot. Now, apparently I'm very good in recognizing these horizontal stripes are not exactly the same. They have like a little curvature. They're a little bit differently in shape. So I recognized that it was the same set of 4 bands just flipped. And then I found the third occurrence of that same blot, as you can see on the right panel in the middle, labeled D and CA. Again, the same 4 lanes with that little dot. So if you look at the labels of these experiments, they're all different. It's the same photo, but it's representing the same 3 different experiments. So we don't know which one is true, but 2 of the 3 are presumably not true. I found another occurrence of a Western blot, the one that you can see that I've labeled with red boxes, that also was present twice. It's the same photo, cropped and stretched slightly differently, but you can see those same, the same bands there, the same Western blots, as we call them. So I realized, I guess, well, 1, I was very angry, and 2, I have a talent for recognizing patterns. And so I've always looked at bathroom tiles and floor planks, as one does, and never realized this was apparently a strange talent I have. But if I go somewhere in a bathroom, I'm like, oh, that tile is the same as that tile, it's the same as that tile, but upside down. This, of course, only works with what we call man-made tile, like human-made porcelain tiles, or floor planks that are laminate. It doesn't work with real wood or real stone, because those patterns in nature are always unique. But when we think about materials that are not nature, like a laminate floor, you'll see the same pattern over and over again. And I don't know if other people always see this. I always look for the same planks, driving everybody around me crazy. Like I see these patterns. And so I thought maybe I can use this for something good, because it's pretty useless talent to recognize, bathroom tiles, but maybe I can use it for scientific experiments and finding patterns like the one I found in that PhD thesis. I also realized what made me so mad is that for me, science is about finding the truth. This is my core belief of what science should be. And the truth, of course, we can have long philosophical discussions about what is the truth. But for me, it's just this simple thing, like you report what you have found. And for me also, science is about building on top of each other's work. As a scientist, we don't, reinvent the wheel. We build on the work that others have done before us. Every science paper cites other papers. We build on the work of decades or perhaps centuries of work that other scientists have done before us. The discovery of DNA or Newton and laws in physics. And we build on that work, we cite each other's papers. So I see science publications as bricks in a wall. We build new layers of bricks on top of the old bricks. If one of those bricks, one of those older papers, contains an error or contains fraudulent data, that means that part of that wall could be tumbling down. It could mean that scientists are wasting their time and efforts and money on trying to replicate work that contains an error or fraud, work that might not be true. And so for me, I think the anger that I felt when somebody plagiarized my work, or when somebody is representing different experiments but using the same photo, that is a deviation from the truth. And so that might lead to other people wasting time and money trying to replicate those results. It might lead to part of that wall tumbling down. And so that is why I do that. Well, science is always based on truth. I think when we peer review papers, we assume the data in that paper is the truth. But yeah, maybe there is some fraud, and maybe there are some errors in science. And maybe we should care about that, because we tend to say science is self-correcting. So I'm trying to play a role in that self-correcting part. So I'm not doing this work by myself. I'm part of a growing community of, we're called like science sleuths, a growing community. And I know that at least one other person in this room is present, a growing community of folks who do what we call post-publication peer review. We look at papers that have been published after peer review, but that still contain errors. And all of these folks here have their own specialty. Some people like myself, we look at images. We look at photos in scientific papers. Other people look at statistical errors or clinical trials or plagiarism or combinations thereof. Some people just look by eye. Some people use fancy computer programs, etc. But so we're all doing slightly different things, but we are a community of folks who care about science and who probably are being seen as troublemakers because we send letters to editors and we want these papers corrected or perhaps retracted. But we care about science as much as I hope other scientists do as well. And so when we find a problem in a scientific paper, we post our concerns on PubPeer. PubPeer is a website where you can do post-publication review. Basically, you can post comments about a paper as long as it has a DOI, a unique identifier, which means that a lot of predatory journals are actually not included. And we also hope that people will install a plugin, the PubPeer plugin, so that if you do a literature search, you might actually get warnings that certain papers have been commented on. As you can see in the left panel, you see I did a literature search. So this is in PubMed, which is sort of the database for biomedical research. And you see these green or even red banners appear. That means that that paper has a comment on PubPeer. So this only works if you have their plugin installed. And the red banner means a paper has either been retracted or received an expression of concern, while the green banner means it's just a comment. And you can click on the green banner and then you'll see the comment that you see in the middle, which happens to be my own comment on a paper by a French researcher. And we hope that people use that. Can I just see a raise of hand of people who have heard of PubPeer before? Not that many, but I hope if you care about the literature and about, what have other people to say about certain papers, that you can install the plugin. It will work with most of the browsers that are available, and it will just sort of play in the background and search for papers that have been commented on. And so I have, I think, around 9,000 comments on papers on PubPeer. How many do you have, David? You don't know. Okay, thousands, tens of thousands probably. So some people are quite prolific, and yeah, we hope to comment on all the papers we've seen. So I focus on photos. When I think about biomedical papers, there's a bunch of ways that people present their data in. So it could be in tables, could be in line graphs, or it could be in photos. This is very specific for biomedical literature. If you think about other scientific fields, a paper might not even have a table or a line graph. It might just have text, or it might only have formulas, or so it depends really on the field. But if you think about molecular biology types of papers in particular, you will see all three of these type of presentation modes. Now, in a table or a line graph, there can be all kinds of problems, but they're pretty hard to recognize. It's actually very easy if you think about it. You can just type in some data in Excel and make, if you are really a fraudulent person, you can make a convincing line graph like that. That's easy to make in Excel. And or you can make like a heat map, or you can present data in a table. In photos, at least, we used to think a photo or it didn't happen. I'm talking about 3 years ago or older papers before generative AI. And I will mention generative AI later on. But before generative AI, let's say 3 years ago or older, we saw a photo and we knew an experiment had happened. Whether or not it's a real experiment, we don't know. But at least with a photo, there's lots of pixels to look at, and we might pick up on all kinds of problems in photos. And like I said, we all have our specialty. I'm good in pattern recognitions in photos. Other folks might look specifically at tables or line graphs. So when you think about photos, there's problems you can pick up on. So I wrote a paper on this, and we sort of divided the types of problems you might spot in photos as 3 types of duplications that you can detect. Now, when a person is really a good Photoshopper, I would not pick up on that. But we do catch the dumb fraudsters, the one who leave traces for us to find. So the 3 types of duplications that you can think of are moving from left to right, a simple duplication, a repositioned duplication, or an altered duplication. So starting with the simple duplication, you see 4 different photos, and the ones that I've marked with these colored boxes are identical. Well, if you look at the labels, they're different. So there should be 4 different photos, but 2 photos are the same photo. Now, I want to stress that this is not necessarily fraud. This could just be an honest error. You're a scientist, you've taken thousands of photos, you just mislabeled them, or you dragged and dropped the wrong one when you made your image for your figure, for your scientific paper, and an error was made. So it's sloppy, but it's not necessarily fraud. We assume, at least. In the middle example, you also see 4 different photos, and you might spot with my colored boxes that I've added that actually all 4 photos overlap with each other. At least they all came from the same specimen. It was just moved under the microscope a little bit around. 4 different photos were taken, but the photos are overlapping. You can spot these overlapping areas. So I spot these by eye, but I'm now using software to help me find those duplications. In the photo, the repositioning could also mean rotation, mirroring, stretching, things like that. In this case, it's just like it was moved around, but there's no mirroring or so. Especially when there's mirroring, when a photo is flipped or rotated, all kind of stretched, we assume there's an intention to mislead. When it's just 2 photos overlapping, that could still be an honest error. But in this case, the person who wrote this paper claimed to have done 4 different experiments, and all 4 photos are from the same specimen. So I don't know, that is pretty sloppy, I think, and that is borderline misconduct, I feel like, but it's hard to prove. The photo on the right, however, we only see 1 photo, but within that photo, we see duplicated elements. This is a photo of a tissue, and it's brown stained for a certain protein. But you can see that within the photo, there's duplicated elements. Now, here we assume an intention to mislead. Whether or not that's misconduct is another question. That's really hard to prove. Maybe the authors were just trying to make the photo look a little bit better, removing a crack or a hair, something innocent. But at least this was done, we assume, intentionally. The actual photo looked different. So this could be misconduct. So these categories sort of are helpful in determining whether or not a person had that intention to mislead. Now, I always like to play the game image forensics. I play this on social media like Twitter or Blue Sky. And when people get the answer right, they can win an emoji award because I have no money to give. But people are very happy when they are one of the 3 persons who got the right answer first. So we're going to play this here, and hopefully folks online can also look at this particular image. And you see 9 different photos, 9 different panels. They're labeled differently. They should all be different independent experiments. But 2 of these photos are identical. And just raise your hand. I'm not going to call anybody out. Raise your hand if you've spotted the 2 identical patterns. This is fairly easy. I think most people should be able to see this. And I'll show the right answer. I hope you got it right. If you see another one, always let me know because I might miss something. So is this, again, this is a simple duplication. It's the same photo, could be an honest error. And in this case, I posted this online and reported it to the journal. It took how many years? 6 years, but eventually the journal issued a correction. And so let's assume this was an honest error. We don't know, but at least this paper got corrected. So, but sometimes it is not corrected. Here's an example of a type 2 duplication. So now we're looking at overlapping images, images that might be rotated, but in this case, they're not. They're in the same orientation. You see 4 different photos, and there's actually 2 pair of overlaps. So 2 photos overlap with each other, and the 2 other photos also overlap with each other. Now, when I look at these images, you can sort of see that 2 images are more similar than the other 2. So just look at the numbers of cells you see here. You might not even know what you're looking at. I often have no idea what I'm looking at either, but you see sort of cells. And the 2 photos that are in the center of the photo have more cells than the 2 photos on either end. And so if you are looking for overlaps, you focus on the pairs that are most similar to each other. That gives you the highest chance. Basically, I think what my brain does at least, is pairwise comparisons. And you look for patterns. Now, just raise your hand if you've spotted at least 1 overlap. All right, a couple of hands there. That's actually very good. So there's 2 pairs of overlapping panels. Now that's a lot, I feel, for, if you're a scientist, you're very sloppy if you produce results like this, in my opinion. But here, yeah, nothing happened. So this paper is still out there. It's not corrected. It's not retracted. Yeah, the authors didn't reply. They didn't care. So we don't really know what's going on. In some of these cases, papers do get retracted. And in some of these cases, they're pretty intense in terms of numbers of duplications. So you see 2 different examples from 2 completely unrelated papers. And I'm showing these to show how editors might respond or react differently to these allegations. So on the right, you actually see the image, part of this image I presented before. It was part of my duplication type number 2 example. It's actually part of a larger image. And here you can see that the PLOS ONE editor, so PLOS ONE is the journal in which this was published, decided that the number of errors were so extensive that this paper should be retracted. So it basically gets a watermark retracted. You can still download it, but it's clearly marked as not being reliable. Now, that means, I think that's actually a good decision. Like, I think if you make that many errors or you do this with an intention to mislead, that paper's just, we can assume that the other data in this paper might also have a problem, might be less visible. But yeah, this paper got retracted. Now, the example on the left, I think, has a very similar problem in it, in that a lot of these panels overlap. But here, the journal said, okay, well, the authors made an error, but we allow them to send in a new set of images, just do a completely different set of experiments, and we'll correct the paper. So here, if you now would download this paper, there's a correction attached to it, and you'll see a completely different figure with images that are very different from the ones that were originally published. Now, in my personal opinion, this is the wrong outcome. This is not a good editorial decision. For me, this is identical to winning the Tour de France which if you don't know, it's a big biking race in France, and then doing a doping test to see if the athlete had used doping during their training, finding that the urine has lots of doping in it, and then allowing the athlete to send in a clean urine sample 2 weeks later. That is not good. And in sports, at least, folks have decided that, you know, doping checks need to be done, and because otherwise everybody's starting to use doping, and need to have consequences. So certain folks who have won the Tour de France have lost their medals because of this. And it seems for me that here the editor did something very different. Like they allowed the authors to send in a completely fresh and new and clean set of images. So I think that's the wrong outcome. But yeah, just to show you how editors can have very different opinions about similar cases. There's no consistency here. Here's an example of a type 3 duplication. So you see two different photos. They don't overlap with each other, but each of these photos has duplicated elements. And just raise your hand if you spotted one. They're much harder to see. And of course, some people are very good at that. Some people have trouble. That's okay. We all have different talents, I always say. But I'll just give you a couple of examples, a couple of minutes or seconds to look at it. For example, if you, let's focus on the photo on the left and look at the bottom left. You see these little orange dots? See if do you see multiple of those? Or if you look at the photo on the right, you might spot in the sort of in the middle, this big hole, like black hole surrounded by orange. Do you see part of that somewhere else? So those are the types of things we look at. So I'll show you the answer. There's lots of duplicated elements or duplicated areas. And the longer you look, and I have a big screen and I play with the contrast, and I can spend the whole afternoon doing this because it's so much fun. And I find these duplications, but usually we only spot one or two and then you start looking more. And there is software helping us find these duplications as well. But the human eye is still so good at finding these things, but you have to spend some time looking at that. But it is a fun, I don't know, it's like finding Easter eggs. I feel it's like a fun hobby. And so in this case, this is the Texas MD Anderson Cancer Center, which is very well, known in the US, but I don't know what the authors did here. I think this goes a little bit beyond trying to cover up a hair or a crack. I think this is really changing the results in a way that is, in my opinion, unacceptable. But the journal didn't do anything yet. The authors didn't reply. And it's just maddening, the lack of response when you find stuff like this. Here's another example. This is actually from Stanford, where I used to work. A professor who produced results like this. There's lots, these are Western blots again. And again, if you don't really know what they are, that's fine, but I hope you can see that parts of these images have been duplicated in a way that is unacceptable, where these lines, these bands, these black dots have been duplicated, or areas beyond, or even moved to different sizes. And it's just unbelievable. And again, nothing has happened. Another result, so this is from a French professor who decided to sue me, or threatened to sue me. He's a professor who claimed that hydroxychloroquine could work against COVID, to treat COVID. His paper was actually tweeted by President Trump in his first presidentship. And lots of people, thought this was, and Trump actually said, this is a game changer, this paper. This is actually not the paper you're seeing here, but the professor produced that. And I wrote a critical review. It's just paper, so I cannot really show you because it doesn't even have images or photos. But I found lots of other problems in his paper as well. So you see some of these papers there, and none of these have been addressed because this guy is trying to sue everybody who, tries to criticize him. And he has gotten away with that for many years. And he tried to sue me. But yeah, he said I was harassing him and I was blackmailing him. I don't know where that came from. But, 3 years, I lived in the fear that if I would enter the European Union, I would be, picked up by the police or something. So luckily that didn't happen. And at some point I even posted a photo of me like, "Hi, professor, I'm in Paris, like come and get me." But he didn't do that. But it was a scary time at first when you get, such a, a lawsuit hanging over your head. He actually doxed me, which means he published my home address online. And so he wrote all kinds of articles about me. He wrote preprints about me. He had YouTube videos about what a bad person I was, things like that. And he had millions of followers and still has, even though he's now retired. And so those folks were starting to harass me. So it was, a couple of fun years, fun being sarcastic here, where I was constantly receiving all kinds of nasty messages on Twitter. I'll show that in one of my later slides as well. So yeah, lots of problems in the papers by this guy. He has published 3,000 papers because he put his name on pretty much anything that left his institution, of which he was the director. Also lots of ethical problems where he did all kinds of research without the proper ethical permit. For example, he asked his female students to do vaginal samples to look at which bacteria they had in their vagina, which is, not something that most people would want to do by themselves. And he said that they would fail their grade unless they participated. Now, without any ethical approval. In the US, this would be coercion, because basically somebody with authority tells you to partake in a medical research study without ethical approval. And so this is just completely wrong, at least in the US. Maybe in France it's allowed, but in the US this would be absolutely not possible. And so we raised concerns. He has now hundreds of papers with concerns raised, all kinds of problems in his papers. These are just the image problems, but there's many other. So some of these papers with problems could be, not very important. They could not really have other papers resting on them like those bricks in a wall. But some papers are actually pretty influential in their field and might result, if there's fraud in these papers, they might result in part of that wall tumbling down. So as part of an investigation with science journalist Charles Piller, who wrote the book Doctored, you see that here on the right, about fraud and other misleading problems in Alzheimer's papers. So Alzheimer's, of course, is a disease that affects pretty much anyone. We all have relatives or will have relatives who will suffer of this disease, and there's no good cure yet. So the hunt for an Alzheimer's cure is a very prestigious one and has led presumably to some fraud. And so we've worked on some of these cases, and you can see some of the headlines there, not just Alzheimer's, but some other research where image manipulation had played a role in even clinical trials, like the papers formed the basis of clinical trials in which patients enrolled, and finding fraud in those papers could have led patients to enroll in the wrong clinical trial, especially in a situation like stroke, where you have maybe minutes to decide which clinical trial to enroll in or have your relative enroll in who's there in the ER. If you enroll in a clinical trial based on fraud, that might cost lives in the end. And if it doesn't cost life, it at least leads to a lot of patients' false hope. So I think some of these cases are incredibly important to talk about. And we're finding more and more of those. So going back to when I initially started this work, I wanted to know how often you would find image problems in scientific papers. So I did a sort of a survey of 20,000 papers that I scanned by eye. This was before there was software. And I published this paper with 2 co-authors who had to agree with me. They were also men, because I thought if you try to say something, as a woman, you're often regarded as a crazy woman and not taken seriously. Unfortunately, this is how some science works. And so I teamed up with 2 men who agreed with me, and both of them had to agree with all my findings. In my set of 20,000 papers that I screened for image duplication, I found around 800 papers with problems. And in 782, we agreed. So I think the percentage they agreed with, my 2 co-authors agreed with, was very high. And we learned from each other. In the beginning, I was a little bit trigger happy, and they were not always convinced I was right. But I think after a couple of months working together, we grew towards a consensus. So that was a great learning experience for all of us. We tried to publish this paper. It was rejected 4 times. Nobody believed us. But in the end, we had to publish it in one of the journals, of which one of the co-authors was the editor-in-chief of. So a big conflict of interest, but this was the only way we felt we could publish it. And it's not published as a research article, but as an editorial or an opinion piece, with clear disclaimers that it was handled by lots of different editors. But maybe we should have done it differently. This is one of the decisions. We just wanted to have this paper out. We felt we did everything we could, and we had it peer reviewed by 10 people and rejected. And yeah, this was how we got it published. So in that set of 800 papers, we assumed that 800, so that's 4% of the total set, that about half of them might contain image problems with an intention to mislead. Now, of course, we don't know what is driving an author. Again, could be honest errors, could be sloppiness, or it could be, just trying to cover up a hair or a crack. But with rotations and stretching or multiple duplications in photos, we assumed there was an intention to mislead. So we assumed that about half of these papers, so 400 of the 20,000, which is 2%, had potential cases of misconduct behind them. Now, we're only looking at photos within a paper. We had no software to help us find duplications between papers. We also didn't look at tables or line graphs. So only looking at photos, this must have been an underestimate. Imagine, for example, if you're a smart fraudster, you would Photoshop in such a way I wouldn't detect it. Just take parts of another photo and put them over whatever you want to cover, and I would not be able to detect it. Or move your sample under the microscope so far that it doesn't overlap with the first photo. I would not be able to detect that. We are only detecting the really dumb fraudsters, the ones who leave traces for us to find. So the 2% of misconduct that we estimated has to be an underestimate. The real percentage of fraud has to be more than 2%. And there's now actually, I'll show some data later, that it might be close to 10%, which is a frightening number in terms of absolute number of papers. It also means that maybe 90% of papers does not contain misconduct. I do want to stress, we're still only talking about that, one bad apple in a beautiful fruit basket of science papers, but there is a significant percentage of fraud in biomedical science. What I found the most frustrating is the lack of response of people, the editors, when we try to report these papers. So that set of 800 papers with problematic images, not all misconduct, but problematic nonetheless, we reported, and about 5 years later, so in 2020, only 1/3 of these papers had received a correction or a retraction. Or you can see that on the left, there's a tiny sliver of orange, barely visible, an expression of concern. But that is only 30%. Like the 2/3 of these papers were unaddressed after 5 years with some reminders, which is frustrating. Now, these were the early days of me doing this work. It wasn't really, I was still considered a crazy woman, I guess, to report these things. I think by now the success factor is just, if you measure a retraction or a correction as success, at least for science, I feel, it's getting better. So now it's about 60% of papers that have received a correction or a retraction. But these papers are reported by now 10 years ago, and 10 years is way too long. These are 5-second retractions, I feel, in some cases. They shouldn't take 10 years. And I understand that as an editor, you need to contact the authors. The authors need to have a chance to report back and say something about it. But it just shows the inability for science to correct itself. It just takes banging on doors for 10 years to get something done. And in some cases, there's still 40% not addressed after 10 years. That's a sad state of affairs. But by now, my work has resulted in 1,500 retracted papers. I've only published 40, but I feel I've left some mark on science, hopefully. Of course, I've also made lots of enemies. This work doesn't go down very well. So I want to switch in the last couple of slides to AI, which I feel is sort of the elephant in the room. If you think about images photo detection of problems in photos, we have to talk about AI, because generative AI can be used for good and bad things. We can use AI to detect all kinds of problems. This is not generative AI. This is more the pattern recognition, machine learning type of AI. For example, I'm using Image Twin and Proofik, which are two software tools from competitors, but they're both pretty good, and they both have their strengths and weaknesses in detecting image problems. Most importantly, these software tools have libraries of images from thousands and hundreds of thousands of scientific papers. So not only can they find problems within a paper, they can also detect an image that has been taken from another paper and reused, because they have millions of images in their databases. So they're very helpful in finding all kinds of problems like plagiarism of images, where people steal other people's images. They can be used, AI can be used for peer review or data analysis. Like, can you find a pattern in whatever, spreadsheet of data you have? So AI can be used for good things. And most importantly, I think it helps folks for whom English is not their first language, like myself, being a better writer. And if you think about it, most of the world English is not their first language. And so it can help a lot of people helping write better papers. And I might even be open to a future where we have AI write our scientific papers. This would have been 2 years ago, I would have said, hell no. But now I think I can sort of see that, where you just give it the data, it writes a nice abstract, it writes a nice introduction, and can stay to the point. It can make sure that it ticks all the boxes, and it can help us write better papers or even peer review papers. But it can also, of course, use to deceive people. It can be used to generative AI can be used to generate fake data, patients that don't exist, data points that have never been taken. And so it can plagiarize papers. You can just feed it another paper, say, rewrite this paper, and in 2 minutes you have another paper that is a complete plagiarism of another paper, but very hard to detect with traditional plagiarism detection tool, because the text is not verbatim the same, it's different. And of course, it can generate data that doesn't exist. So you might spot, again, the dumb fraudsters who have left a trace of generative AI generated text, like, as an AI language model, I cannot help you with that. If you publish a paper and are dumb enough to leave that one sentence in, we know that probably the rest of the paper was also not written by that author. Or you can publish a paper in which you produce the rat with the giant penis that did the round about a year ago on Twitter. We had a lot of fun with it. And, this is not a biologically or physiologically accurate representation of the anatomy of a rat, as you can see. And but, this was AI generated. It was disclosed by the author, but the text was also AI generated. And so the paper is now retracted. But yeah, stuff like crap like this is doing the rounds and is getting accepted and is getting through peer review. So there's just an enormous amount of AI generated paper. And as scientists, we all feel this pressure to publish. And so it's very tempting to use AI to generate our papers. So again, you can see AI generated text. And there's certain words that are very popular by ChatGPT, like delve. And if you look for the frequency of the word delve, you see strange things happening. You see basically a hockey stick curve where, since 2022, 2023, there's an enormous increase in the word delve. And so you can do all the kinds of linguistic analysis to show that a lot of people are using ChatGPT to generate their scientific papers. Now, some people will say, but I love the word delve, and I've always used it. And so you don't want to falsely accuse folks of having their paper written by AI. But if you do these type of meta-analyses where you generate data on thousands or millions of papers, you can see these patterns. And we can conclude that probably a lot of papers nowadays have had some help or all the help from ChatGPT. Is this okay or not? Well, we can discuss that, but I think I'm most worried about images, about data generation by AI. Maybe we shouldn't worry too much about text, but how about data? How about images? Like I said, we used to think if we saw a photo that an experiment happened, but now it can generate convincing photos that are not real. For example, if you have access to Photoshop, you have now access to their generative fill feature, where you can feed it a photo of these are mouse or rat tumors. And Cheshire, who is one of us sleuths, he was trying to prove here that generative AI is pretty good in generating photos. If you give it a photo of something real, it can actually fill part of it or like extend that photo in all kinds of directions. So in this case, the right column, those 4 tumors on the far right of the left photo are fake. Those are AI generated based on what AI saw in the other photos. Now, if you just take those 4 tumors on the right and would publish them, our software that would not be able to find it because these are unique photos. And you can do this over and over again. You can, have 10 times use that same photo and have 1 extra column, use only that extra column, merge those in some ways, and you have unique data that we would not be able to detect as fake. And it's really hard to look at a photo like this and know if it's real or not. I would definitely not be able to do that. You could do some fancy analysis on it, but it's really hard to find the problem here. And similarly, you could also train. This is just ChatGPT or Photoshop that generated this. But you can actually also train models on real photos. In this case, photos of tissues. And if you feed it enough photos of tissues like lungs or hearts or kidneys, you can actually have your model generate really convincing photos that look pretty real. I would not be able to tell which photos are real or fake. The top row actually is real. The bottom row is fake. So again, these are unique photos. Every time you ask the model to generate a photo, it will generate something new. And so this is tempting. If these technologies are so easy to use, I'm pretty sure they're already being used by fraudsters to generate data that doesn't exist. And this brings me to scientific paper mills. So paper mills are sort of scammers who are selling fake or low quality papers. Well, actually, they're selling the authorship positions. So these scammers advertise, for example, on Facebook. You can actually go on Facebook and search for these call for authorships. These are usually paper mills. They promise you to be an author on a scientific paper that you didn't write yourself in return for some money. You just pay them some money and they'll put your name on a paper. You want a paper about microbiology or computer science, whatever, we have it. And so they sell these authorship positions. And we can all appreciate as scientists that we feel this pressure to publish and be even maybe sympathetic for people in countries that don't have a lot of money for science, who are looking for a better future in another country. Maybe they want to do research in New Zealand, or they want to do research in Europe, or maybe not so much in the US right now. But you can see that people are hoping for a better future. In order to do a postdoc, they need a resume, they need some papers to have published before. And so here is an easy way to find, to boost your resume, to add some papers to your resume that you didn't write yourself. And so these scammers, these paper mills, use all kinds of methods. It's not just one paper mill. There's many of them in many different countries, many different forms and ways they use. For example, they could plagiarize a paper, or they could synonymize all the text in there, or they could use AI now, of course, to generate very easily a review paper that we don't really need because it's a paper on a topic we already have millions of review papers about. But they can generate in some way all these fake papers. They use templates, they use hired writers, they might steal photos, they might generate them with AI. And it's now estimated that there's the number of paper mill generated papers is extremely on the rise. You can do linguistic analysis. You could do, like in this paper by Reese Richardson, published recently in PNAS. he analyzed the amount of paper mills. And you can see this is a log scale of number of articles. You can see that the number of paper mills is growing at a much faster rate than the number of papers. And there was also a recent preprint by Jennifer Byrne in Australia, where she estimated about 10% of papers in cancer research might have been written by paper mills. This is based on linguistic analysis. And so the number of paper mills is extremely on the rise. And it's really hard to stop it because at first glance, they look real. And how do we prove that they're fake? So that is actually a something we can worry about. So I want to briefly mention that maybe we shouldn't try to detect what is fake. Maybe we should try to authorize or authenticate what is real, which is sort of, turning things around. Maybe we should have every image have a sort of a watermark to prove that it was really taken by a camera or a scanner in a lab on that day, and know what types of manipulations were done to an image. You can see, so this is just one example, C2PA. You can see that newsrooms like BBC or the New York Times would also really want to know which photos are real or fake. And so this not only plays a role in science, but as we see the growing amount of fake photos on social media, we need better ways of knowing that something is real. And so I think this is something we need to invest in. And so I want to also still show a slide with some of the harassments. I already mentioned this, the French doctor who made all these videos about me. Because if you're thinking about doing something like I'm doing, like let's criticize some papers, it's not something actually I would recommend as a career choice, especially if you're early career, because you're going to create a lot of enemies. So you have to be somewhat resistant to criticism, like the emails I'm getting, the hate I'm getting, and the lawsuits that some of us folks are getting as well. So it's not a career choice I would really recommend. It's probably something for later in your career when you don't really care about having a career. But we've heard of whistleblowers who are early career. Let's say you're a grad student, you're in a lab where a bullying professor is asking you to Photoshop something, or maybe not even asking that, but say, if your results don't work, I'll find another postdoc who can do the work. I think in a situation like that, you might be tempted to fake your data and please the professor, because you'll lose your job if you don't. And especially if you're on a visa, that might be, harmful for your career. But I've also heard from whistleblowers who have raised concerns with the authorities at their university, and then suddenly their key card doesn't work, or their parking permit is not extended, or suddenly their grant is ended, but then, they have to leave the university. So you might be seen as a whistleblower as a troublemaker, as somebody who sort of needs to leave the university. And so this type of whistleblowing is really, really hard. It shouldn't be. We should all embrace criticism, but we don't, of course. And universities very often choose to protect the professor who brings in the money, the grants. And a graduate student who raises the alarm about certain ways that the research is being done might be seen as the troublemaker. And so this type of work is tough. I'm not, I can talk about this, but there have been nights where I was crying about emails I was getting or lawsuits. I mean, they're really, really scary for an individual without a job like I have. You don't have any protection. So this type of work is tough. This is, of course, while some of us choose to remain anonymous, and you can post on Papier anonymously, which is the route that many people choose. I do think more and more, there's more and more, positive vibes about what we do, more appreciation about what we do. So luckily, a lot of publishers are starting to work with us, but universities still don't really like this type of work. So I'm always happy when a university invites me and allows me to talk about this, because there's a lot of conflicts between what a university likes, their reputation. We don't do any misconduct here, but it happens everywhere. And I feel that universities should be open for that. And criticism like this done by professors at universities, I mean, that's horrible. Like, I criticize his work, not his personality, but he, this guy in France had all his followers harass me and is still writing preprints funded by French research funds about what a horrible person I am. And that is just wrong. Like, why is this still happening? So with that happy note, I will end and sort of remind everybody why I do this work. It is because I feel science should be about finding the truth. So I like to play a role in that. The rewards for misconduct are high. If you do misconduct, your results look so much better. You might have that paper or that citation that you need for your career, but the consequences are low. There's very few papers that are being retracted. There's often very little consequences for the folks who are caught, especially if they're, a professor. If they're a grad student, they're seen as disposable, but the professors often can, you see decades of work done by the same professor with different graduate students, but there seems to be in some labs this culture of misconduct, and nobody seems to be able to do something about it. I've talked about the mass production of fake papers by paper mills, some of which, and probably more of this, is aided by generative AI. How we can probably only fix this by focusing less on quantity. But as scientists, we love metrics. We love to look at how many papers did a person publish, how many of those, how many times were they cited, how many were in Nature or Science. We love to look at metrics like that. And we like to, when we hire somebody as a faculty member, we love to look at their resumes and do these types of comparisons. And it's very difficult to find other metrics. So we love to focus on quantity, but how do we focus on quality? That is much different. And it's easy for me to say we should focus less on metrics, but I realize how tempting it is. We all love top 10, researchers, or we all love these little lists. And so as long as we love these lists and we focus on quantity, we're maybe sort of encouraging people to cheat the metrics. But it takes the role of everybody involved. It takes not just the role of graduate students raising the alarm, of folks like me yelling that we should do things better. It takes university leaders to make change. For example, there's universities who require their grad students to publish 4 papers to be able to do their PhD. Now, in my opinion, when I did my PhD, if I had 1 paper published, that was good enough. You worked your butt off for 4 years or 5 years. You had, most of the experiments still failed because you're young and you're still learning it, but it was okay. I had 1 paper published, I had a couple of submitted, and that was enough for me to do my PhD. But I think as we raise the bar and allow and require grad students to have so many papers published, we might enable cheating in some way. We might encourage it. So I feel as grad students, maybe we should think about having grad students replicate an older paper from another lab, and that should be enough for their PhD. Maybe, of course, they should do some other and novel research, but have at least that 1 paper, no matter what the outcome is, might be positive or negative, but have that paper published. And these are changes that only university leaders can make. And then finally, the tremendous cost of misconduct. I do want to end with that. It's not just the cost of scientists who are trying to replicate results that might have been fraudulent, which could lead to years of failed research. It's also on a secondary level, the cost is for folks like me who do sort of post-publication review, the harassment, the lawsuit, the criticism. That is, the cost for us personally. But then finally, on a third level, the cost for science as a whole. So we see all this misinformation on Twitter, and it's very easy to leave this room or this talk thinking that all science is flawed and we should give up on science. No, that should not be the message of my talk. The message should be, yes, there are problems in science. We need to fix that, or at least be aware of it. But we should also appreciate science, because we need science to fix the big problem in the world. We have climate change, pollution, hunger, we have pandemics. We need science to help us fix these problems. But we also need to know which science we can trust. So that is where I try to play a role in it. Thank you. Let's see. Oh, did anybody? I have no sound. I have no sound. Okay. That's not good. I hope anybody heard it. All right, so there were 7 messages, but there were about people who couldn't hear anything and then they could hear. So that's not good. Are there any questions here in the room? And then I'll also invite folks online to submit any questions there in the chat.
I had a question about peer review.
Right.
And do you think peer review system is working? Because it's obviously missing potential.
Mm-hmm. Yeah, so the question for folks online is peer review broken? You know, should these things be detected by peer review? And so it's hard to answer. I feel that peer review is a volunteer job, at least in the current setup in the, you know, the way we do science. It's often a volunteer job. It's not paid. It's done by scientists who probably spend some Saturday morning or Sunday afternoon doing this work, unpaid and unloved in a way. But we feel it's all, you know, part of our duty as scientists. But I feel that, so some problems should have been, I feel, detected by peer review. For example, I've found a couple of papers where prostate cancer, if you look at the table that showed the characteristics of the patients who were investigated in this prostate cancer research, half of them were women. And you're like, okay, that's unexpected. And this is it was part of a paper mill where papers are written on a template where, you know, one molecule is changed by another molecule. It's all fake data. They change one cancer by another cancer, and they forgot to take out the women in this particular paper. So that, you know, a prostate cancer peer reviewer should have picked up on basics like that. But I think in a lot of other problems, like image problems or table problems, as peer reviewer, you're not really looking for fraud. You're not fraud detecting. You're assuming that the data you're peer reviewing is real. So there's this trust that we tend to have in science. So while some of the problems I feel should have been detected in peer review, during peer review, I feel that real fraud detection should be done by publishers. We pay publishers a lot of money, whether it's on the open access model or the subscription-based model. And so I feel publishers should add some of that quality control. It should be done by professionals. I don't want to work full-time for, you know, I don't want to have a boss, but like folks like me who can see these problems or know how to operate the software should be hired to scan, for example, papers once they have been accepted. Do an additional fraud detection round, if you will, and not just images, but have ethical approval been done? You know, are the animal experiments done in an ethical way? Is the statistical analysis done correctly? These are all things that peer reviewers often cannot do themselves, and there should be additional rounds of quality control by publishers. Now, publishers will then claim, but that will make scientific publishing even more expensive. Well, they make a lot of profits. I think scientific publishing is actually the industry that makes, I always forget the detail, but like 30–40% profit. Like it's very high. I do think they have some money to hire a couple of extra folks to do this work.
Elisabeth,
yes.
Do you have a sense of which fields of science are a little more problematic?
What fields of science are the most problematic? Well, I've been talking about images, about photos in scientific papers, and my background is in molecular biology, microbiology. So the type of images that I'm looking at are images I'm familiar with. So the fraud we find are mainly in molecular biology papers with Western blots or photos of cells or tissues and things like that. Now, other fields might, like I said before, might not have photos. They, you know, think about mathematics papers that only will have formulas or so. And I'm looking at these papers. I have no idea if the formulas are correct or not. They're just formulas, and my brain cannot handle them. Or if you think about philosophy papers or economy papers, they might look very different. So I'm assuming there's probably fraud everywhere. But the images are the most visible part. So because we love images, we can see problems in them, at least in older papers. Most of the comments on Papier are in molecular biology, oncology type of papers, yes. That doesn't mean there's no fraud. It just means we need more experts in those fields to scan the literature and raise problems if they see them.
Yeah.
Yeah.
In mathematics, you mentioned I had the same issue as well. So, for example, I read a paper. Mathematically, it was 100% correct. Everything was fine. But when you were going to replicate the results, it didn't work. Right? I tried to write the code to replicate the result. I couldn't get the same result.
Oh.
So I contacted the person who wrote the paper, and somehow I found the code that they wrote, and the code was different.
Oh.
So they claim something,
Right.
but they produce something else.
Oh.
So it's very difficult to detect something in mathematical.
Right.
use,
Right.
because you don't have access to the code, the
Right.
running part. You know, you can see the mathematical. It was okay, but how they did get the results, we don't know.
And do you think it was just some honest error, or it was like deliberately?
It's impossible, and actually it's happened because I started my postdoc based on that paper. I
Right.
said, okay, they published that paper,
Right.
so which means it's
Exactly.
global, right? So, and my idea was based on that.
Mhm.
So that was the point I started to replicate the results for myself first.
Right.
To
Right.
get the idea what's actually going on, and then create my new work. But I realized that, okay, it's not gonna work. And then I proved mathematically why it's not working. And I presented proof to the professor I was working,
Mhm.
and she wasn't agree with me.
Mm.
And we had about, we wasted about a year discussing about this stuff. Now she realized that, okay, it wasn't
Mhm.
correct, and I
Mhm.
showed the code. This is not the same as what mathematical parts saying. So now this has happened, but I actually, that was a good thing. I was going to send an email or something to the journal, say, okay,
Right.
this is not the right paper. This is something wrong in this paper. But
Yeah.
as you mentioned, because the peer reviewers, they don't have access to these details,
Mhm.
so they can't see.
Yeah.
these.
Right. And it's an excellent example of folks wasting a lot of time, and you know, there's so much frustration about trying to replicate papers that contain what appears to be maybe a deliberate error or deliberate, that's too nice, but yeah, you know, like an intention to mislead perhaps. So in your case, I would actually be I'd probably give you the advice to write a commentary on Papier under your full name or not. If you dare to do it, depends on your position and vulnerability. But if you feel like, what the heck, somebody else needs to know about these problems, write your analysis on Papier. And maybe to be very nice, you could write the author again and say, I'm going to publish this on Papier. And, you know, can you work with me or not? And probably they don't want to comment on it, but this invites them to have an open and public discussion about this, and warn other people not to waste, you know, a year of their lives trying to replicate the data. Yes, another question there.
And one of your first slides with that I think started your interest was a PhD. Did you consider contacting the university about that?
I think I did. It's so long ago. I wrote to the are you referring to the plagiarized article or the images? So those had been published also. So I didn't write to the university, I think, but I wrote to the editors because the chapters of the PhD thesis had also been published as publications. So I don't necessarily care too much about PhD theses anymore, but I care about published papers that are publicly available because other people base their work on that. And so I wrote to the editors of the journals, and both papers got retracted. But I don't remember if I wrote to the university, to be honest.
You mentioned a number of very highly regarded places like MD Anderson and, you mentioned Stanford, etc. And one would have hoped that the universities such as that and centers such as that have sufficient ethics and integrity to be able to deal with.
One would hope. I mean, there was even the Stanford president, who was not the author on the paper I showed. It was a different professor, but the Stanford president was had also several papers on Papier, and a journalist who happened to be a first-year undergraduate, 17 years old, at Stanford University studying journalism, wrote about that case. And in the end, the Stanford president had to step down, but there was no misconduct, but he had to step down. And so you can see how universities are actually not investigating these cases very ethically or honestly. They actually wanted to investigate it first by their board of directors, of which the president is the boss. So what's the outcome going to be? So I was one of the persons who said, that is just completely bonkers, because there's no way that board of directors is going to say that their boss is guilty of misconduct. I feel this needs to be, in this particular case, needs to be investigated by an outside committee, which in the end happened. There were lots of lawyers involved. And so again, of course, the outcome was that there was no misconduct. And it's really hard to look at these papers with duplicated images, and, these things don't rhyme with each other. Like, there was misconduct. I don't, know who did it. I don't really care perhaps about it, but these papers should be retracted. And so it's really tough to get universities to be investigating these cases. If it involves a grad students, sure, they're thrown under the bus. But if it involves professors or even presidents, it's very rare that these cases are investigated in a way that, that feels like it was done honestly. There was an immunologist in China, like a top immunologist, who I investigated with 50 plus papers on Papier. I posted all of them. And then people were saying to me, oh, this is like the top immunologist in China. You'll be, regarded as a persona non grata, because he was like an academician. So he was like part of like the top tier in Chinese research. And again, the Chinese Ministry of Education did an investigation and concluded there was no misconduct. And so all my haters say that I was wrong, because the Chinese government investigated and, Elisabeth Bik was wrong. I feel I'm right, like there was misconduct and he was responsible. His name was the senior author on all of these papers. And so there seems in very many cases like folks at high positions are being protected by their university or by their country even. And it's really hard to actually do something that is good for science. And so there's too much conflict of interest in most of these cases, unfortunately. I'm going to see if there's a question online. Just to make sure, how do I see that? Oh, okay, so there's a couple of questions. Let me. Okay, thank you so much. Great talk. Multi-layered question. Okay, I'm very bad at those. We'll do them one at a time. Have you identified fraudulent images during the peer review process? You notifying the editor. So I don't really so the papers I've peer reviewed usually are in my own field, like microbiome research. They often do not have photos. They have line graphs, like heat maps, ordination plots. And it's really hard to detect errors in that. But there was one case where I peer reviewed a paper and I'm like, that data looks so beautiful. That cannot be true. I've never seen such a beautiful ordination plots where all the patients are here and all the healthy people are there. That is just not reality. Usually there's an overlap of data. And so I did contact the editor and asked them to ask the authors to send in the raw data. And I did the ordination plot myself in R, and I programmed it, and it looked completely different than the published data. Like, I think this data is made up. And so the paper was rejected. I'm sure it was published somewhere else, but I did reject it for that reason. And I've heard of other people who said, oh, thanks to your image challenges online, I'm actually looking for these cases, and I found duplications during my peer review, and papers were rejected because of that, or corrected. But yeah, so but even if they're rejected, they'll find another home somewhere else where the peer reviewer is not paying attention. Next question. If so, do you tend to get better results discussions when the fraud is identified in the peer review compared to identifying it post-peer review? It's fascinating that so many published research gets flagged and then left for so long. So it's really hard to know what happens before papers get published, because that part of the scientific process is not visible. It's not public. Peer review usually has confidentiality issues. So occasionally, like I said, I'll get emails from or direct messages where people actually ask my opinion about images. And I know that some journals are starting to use software like Image Twin or Proofic and finding these problems, but we still see too much, overlapping or photoshopped images being published. And of course, AI is probably now taking over, how to do fraud. So I do think it's much less work for during the editorial process to flag these problems. Then once a paper has been published, it's much harder to take it down or to at least flag it, because data, especially a couple of years later, data has been lost. While when a paper is submitted, the data is still warm, so to speak. The original data is all there. And if there's any questions, that is the best time to address it with the authors, as opposed to paper gets published, 10 years later, we discover a problem. Now the data is lost. So in terms of much more effort should be put into quality control before a paper gets published. The next question is, how do you determine what to include, how to phrase papier comments? Would you encourage anyone who identifies integrity issues to comment? So papier is moderated. You can either comment under your full name, or you can create an anonymous account. So you will get a password. There's no IP tracking, so it's really anonymous. Nobody can subpoena papier and say we want to identify who that user is, because they don't have that information. So you can comment completely anonymously, but you cannot just say whatever you like. And so a lot of folks who have never commented before find it hard to comment on papier because it doesn't come through the moderation process. So I'm out of moderation, so I can post what I want, but if people still want to flag it, they can flag me. And my comments are sometimes taken down because we sometimes do it a little bit too sarcastically. But it's, you cannot say you're a fraud. You cannot say this image is photoshopped. You have to use more legally, sensitive wording, like this image looks remarkably similar to that image, or this image appears to overlap. Like, it's your opinion. You're right, but it's your opinion. And this is all because of legal issues. You cannot accuse a person of being a fraud. You definitely cannot say that. So there's moderation rules, but yeah, you can see what people post on papier and what gets through moderations. Then you can sort of see what language to use. But there are certain rules where papier seems to be a little bit too strict. For example, if I find a paper with 30% of the citations from the author themselves, I think that is worth flagging. But now they have a rule that it has to be more than 60%, which I feel is completely bonkers. Sorry, papier folks, if you're online, but I think that should be 10%. But there's discussion about like what is reasonable, and it might depend per field or might vary per field. Yeah, there might be situations where you're the only expert on that topic and you have published all the papers on that topic. So you'll cite yourself only because nobody else knows about that topic, maybe. But that's, at least in my field, very unusual. So yeah, look at what other people post and there's a frequently asked questions about how to phrase your questions. Thanks very much, Elisabeth. As a French scientist, I've followed closely your battle with that professor and his team. It's unbelievably what you have had to go through just for standing up for science. Couple of questions. Is it worth the effort trying to correct things or flag papers published in predatory journals? I think it's worth the effort, but I try to focus on things that have been published in more reputable journals. Very often, predatory journals do not have a DOI that works. They make up DOIs that don't work. So if you type in the DOI, you cannot create a papier comment because the paper is not in their system. So if a paper is not in their system, it's not worth flagging. It might be part of a larger set. So sometimes I do send them to the editors or to the institutions, but I don't focus on it, but I wish, because I have only a limited bandwidth, so I choose the ones I feel are more in the mainstream literature. But I would love hundreds of people to join me and fight those for sure. So it's worth the effort, I feel, but I'm focusing on other things. Do you have examples of false positives where it looked like alterations, but it wasn't? What can authors do to defend themselves? Yeah, this sounds very, how do you say, cocky, but I feel when I flag something, it's a real duplication. I sign things with my full name. Now, sometimes a duplication within an image, I mean, it's very hard, I feel, to have a false positive, but occasionally we find problems, what I call corner cloning, where you have a photo and in the, let's say the top left corner, there's the letter A, and then there's a panel B, panel C, D, etc. Now, sometimes authors send in these photos with A, B, C, D, but the font of the letters is not the same as the journal likes to use. Like you've used Arial, they love Times Roman. And so we've seen cases where the journal actually is taking, like they take a little part of the tissue and patch that over the A and the B and the C, and then add their own font A, B, C, or they don't like the bottom left, it needs to be the top left or whatever. So we've seen cases of corner cloning where all the top left or bottom right or whatever corners have been photoshopped. Now we know this is very often done by the journal, but we still feel it's worth raising because very often the authors had no idea. And a journal shouldn't be doing that. I mean, you just patch a black box then over it or whatever. Like, but yeah, so are those false positives? I feel they're still worth raising the alarm over, but in Western blots, I think in the beginning, I did make mistakes. There's a couple of images where images are the same. I'm pretty sure about that, but are they inappropriately duplicated? Like maybe they're actually the same experiment. So there's cases where I occasionally am wrong. It is a duplication, but it's inappropriate. But because I post under my full name, I have to be pretty sure about what I see. And so I might be wrong, but it's still duplication. It's maybe an appropriate one. Is there a question in the room before I go back to online? I don't know how far we still have. But that was actually the last question here. No other question. I think we're 2 minutes over. So, well, there's no more questions. Thank you so much for hosting me here. It was my pleasure to talk for you today, and I hope you I don't know, don't go home telling that all science is bad. I hope you are even more passionate about science than you were before this talk. Thank you.
Thank you all for attending this presentation today, everybody here in person and those online. Elisabeth, I thoroughly enjoyed your presentation. I enjoyed your enthusiasm and enjoyed your source of enthusiasm, discovering the chief and identified duplicate images. The examples that you showed of duplicate images were interesting and challenging, and they show that you're very talented at picking. I couldn't pick some of those things that, you know, some of they were
batting faces.
You're very, very talented, and thank you for your hard work and for enduring the challenges of the profession, the litigation, and not receiving any Christmas cards and all those types of things.
Well, who does nowadays? Nobody sends them anymore.
That's it today. Thank you all for attending. I'll close us off. Kia tau, kia tātou katoa, te atawhā ko tātāre ki wahiu karaiti me te aro, te atua me te whiwhinga tahi kanga ki te wauru tapu ake āmeni. Thank you.
Thank you.